What is our department? Forest resources. I have some FMI, but it's forest resources. Welcome. Thanks so much for joining us. I'm Ingrid Schneider from the Department of Forest Resources, and this is our third and final fall seminar for the Forest Resources Seminar, where we inspire integration and innovation. I'm delighted to be able to introduce our presenters today. They'll be collaborating in this presentation, and they are collaborators. And we have joining us Drs. Karwan and Rose. So you probably read their bios in the announcement, but just a couple of highlights from them. Dr. Diana Karwan is an assistant professor here in the Department of Forest Resources. She studies hydrology and watershed management, which you probably know. What you may not know is that she was actually afraid of rain and thunderstorms, which may have driven her to study hydrology, who knows. And she also has a collection of Lego minifigures of the difficult to find women geoscientists and NASA figures. Women NASA. Women in NASA.
Women of NASA.
Women of NASA
just got released and sold out.
That's right. So if you're looking for some valuables, check her office. And she'll be joined today by Dr. Lucy Rose, who is a postdoctoral person here in the Department of Forest Researcher, person, I was trying to think of the word, here in the Department of Forest Resources. And she also focuses on hydrology, but also biogeochemistry in forested watersheds. And something, you probably know that about her, but something you may not know about her is that she has been an English teacher in Japan, a science teacher in Pennsylvania, and a fire ecologist in the northeastern part of the United States, or eastern part of the United States. And she's very happy to be doing biogeochemistry here in the Midwest. So I'd like to welcome them both today to talk about the complications of the relationships between land-covered management and surface water, and also remind you that our seminar series will start back up at the end of January. January 31st, we'll be hosting Mike Kilgore to talk about value and pricing. So with that, Dr. Diana.
Thanks. So Lucy and I are going to kind of give you a 1-2 punch here on our work that all surrounds measuring this kind of complicated relationship between land cover, land management, and surface water quantity and quality. And so to kind of start this off and set the stage for this, I would like to share with you, I had the experience that most kind of adults in Western civilization should have once a year by going to the doctor and getting an annual checkup. And as a part of this, they took blood out of my arm. Now, I was slightly upset because I had to fast for a really long time, which meant no breakfast and no coffee. But what the doctor wanted to do was get a series of vials of blood out of my arm and run a bunch of tests on them, right? So they can check a bunch of boxes to look at white blood cell count, cholesterol, etc. And they're using the chemistry of that blood to infer something about how my entire system, my entire body is functioning. And so this is what Lucy and I and our lab group kind of take this approach to looking at streams and surface water. Right? And so whereas in the cartoon stock cartoon, the doctor's like, what's your blood type? And the patient says red. So if we're going to place that analogy in a river, we would say, what's in the river? Well, water, right? Of course. Water is made up of hydrogen and oxygen. Lucy and I and many other hydrologists, biogeochemists, including those that we work with, say, and there's all kinds of other chemistry. And we can use the chemical composition of the water and other particulate matter, sediments, floaty stuff, leaves in the water to tell us something about how that entire watershed is functioning in a unit. How resilient is it to change? What's a pressure point that might drive a level up or down? And so this is kind of the framework that we're using to take the pulse of watersheds. Moving forward. So we're going to highlight 2 example kind of field sites linked to watershed case studies where we work. I'm going to be focusing on the White Clay Creek watershed in Chester County, Pennsylvania, where we have a really kind of long-term, long-standing sample set and data set that I was a big part of collecting prior to my arrival here at the University of Minnesota. And Lucy is going to focus on our work in the West Swan River, which is in St. Louis County, Minnesota, just south of Hibbing. And this is work that's really been going on for the past 2 years and funds the majority of Lucy's postdoc. So we're starting with White Clay Creek that has a lot of ongoing data collection we'll hear about in a minute. The big picture questions here are looking at water and material in the water. So you can probably see that this is when the water is flowing over the road and out of the bank. So this isn't a flood. We're moving a lot of what looks like brown water because it's carrying sediment material with it. What exactly is exiting the watershed here by our gauge? When does the landscape, the land use matter for what's coming out of the watershed? And when does the landscape play a backseat driver to processes happening in the river channel itself? We really do have an unprecedented, and I haven't added up the pricing index, Mike, of the data that we've collected in White Clay Creek, but it's well into the millions of dollars in terms of the chemical analysis and the labor that's gone into this. So this landscape looks a bit different from what Lucy will present to you up north. It's a small watershed, about 7 square kilometers, and the land cover really is about 2/3 to 3/4 agriculture. And this is low intensity cropland. It's nowhere near the tile drained mass production that we see in parts of Minnesota. And about 1/4 forested, as shown in the green. So what you might notice is that these green patches really are running right along the rivers, as shown in the blue lines. This is an area of the country that's experiencing net afforestation. There's been a lot of stream corridor and riparian forest plantings that have been done since the 1970s approximately. So we now have 40 plus year old kind of mixed hardwood forests in this area. So if we look at the trajectory of agricultural land, it's going down in the mid-Atlantic, and the area in forest is going up. And what we want to know is from a landscape that's mixed with some row crops, mostly corn, pasture area with a lot of horse farms, and we have these really deep stream banks, pretty tall. What you'll notice here, this is one of the undergrads that was working with me when I was at the Stroudwater Research Center, and the stream banks are about as tall as an adult female. So I needed a ladder to get in and out of the stream from the stream side riparian forest. What contributes to what's coming out of the watershed that we're sampling in bottles at that one location at the outlet? And this is what that stream would look like not under the flooded conditions. So we really want to know what gets here, what's making it up, where did it come from? So we take a stream, for example, this one in base flow, and we sample the bejesus out of it, right? So thank goodness my doctor didn't take that many vials of blood coming out of my arm because I probably would have passed out in the doctor's office. So we take a lot of water samples in anything from 1 liter bottles to 55-gallon drums, and we run many chemical analyses on them. So this is just to give you a sense of the deep chemical menu. There will not be a chemistry exam at the end of this talk. But to us, water in bottles analyzed for chemistry means lots and lots of data that we can use to answer our questions. So in addition, I should mention, in addition to measuring the water in the stream, we also do some water collections from groundwater wells, from soil water lysimeters, and one of my main interests, particularly precipitation. And so what happens after we've gone through this whole menu of analyses? We'll focus on a few things here. So this is what the stream would look like in White Clay Creek if you're, say, hiking in some of the forested area around it that's all under conservation easement. But what Lucy and I end up kind of seeing in the back of our minds is what is graphed in data. So what we're seeing is a very characteristic plot to hydrologists, where we're looking at some metrics of the water and what's in the water over time. So you'll see a lot of these plots called hydrographs, where the x-axis is always some metric of time. So here we're taking an example of August of a single year and going from August 1st to August 31st. Kind of hanging down from the top is what the precipitation is doing. So when this line, the blue line, comes down further, that means we're getting some rainfall occurring. So precipitation is coming down onto our watershed. Across the time on the bottom, we have this light blue line representing stream discharge. So this is very, very broadly the amount of water coming out of the stream at this area where we're looking. And what you see in other bits of data here, what we'll often focus on is the suspended sediment in the data, measured as total suspended solids. So we have measurements of this taken in those bottles in the black asterisks. And then we have ways of predicting what that would be as a continuous measurement across time, as well as some other information taken during the storms here. So one thing you might notice is it looks like the river is relatively low and then it jumps up and comes back down, jumps up and comes back down. That's usually happening here in summer in conjunction with when the rain is coming. It rains, the water level in the stream goes up.
Hey Diane, just a quick question.
Yeah.
Why such a strong spike on the 28th?
On the 28th, so this was August 28th, 2011. So I did not plant that question, but thanks, Mike. That really helps get at where we'll be going. So for the 2 to 3 year period of really intensive sediment chemistry sampling coincided with the fall of 2010 to well into the year 2013, which is when I moved here. During that time, we experienced Hurricane Irene and Hurricane Sandy hitting the mid-Atlantic of the United States. And so Hurricane Irene is what hit on approximately August 27th to 28th of 2011, which is the month we were looking at. So when we get a hurricane, the water level goes way up in the stream. It was kind of, fortuitous for the sampling regime that a little over a year later, we got a second hurricane. And so what we're able to do is have multiple extreme events to be able to think about when we push this watershed system really out of its box, push it out of its comfort zone, what does that chemical signal tell us about the functioning of that watershed? Will it return back to normal, meaning prior condition before that hurricane? And what happens during that hurricane? And so one of the things that we use chemistry to help us tease apart is to be able to compare where the water in the stream is coming from during that hurricane. And we did see a big difference. So I'll point, here we're looking at some of these same time series plots again. The rainfall coming down from the top. In this case, the line is black, but it's the same type of plot. We have these big spikes because now we're only looking at, say, a 3-day interval around the time of the hurricane, where the blue line is the stream water amount coming out. However, this red dotted line that's underneath it is representing, based on what chemical analysis tells us, the water that was stored in the watershed prior to the hurricane. So this is not rainwater that's coming out the bottom of the watershed. This is what was in soil water and what was in groundwater. And one of the things I really want to call your attention to is this red line is pretty far down there for Hurricane Irene. But Hurricane Sandy that hit about 14 months later, it was right around Halloween when Hurricane Sandy hit. It was like Frankenstorm is what all the news media called it. This red line is a heck of a lot closer up to the blue line. So even though we're getting the same watershed hit by a hurricane, so a pretty darn big storm, the components of the water that are coming out of the bottom of the watershed are different. And we likely attribute this to watershed condition at the time. How wet was the watershed when the hurricane actually hit? In addition to looking at the source of the water, I love sediment. So those of you that know me know that I love seeing waters run really muddy in rivers and get really fascinated by where this comes from. I don't know. This is what happens when you study what you're afraid of. You go way over on the deep end of something else. So looking at many, many years of data where we're looking at one water quality metric taken out of all those bottles of total suspended solids. So this gives us about the concentration of the floating particles in the water. So this is one of the things that can make the water brown. What does 10 years of data plotting that sediment concentration up the y-axis and the river level discharge across the x-axis, here in log space because the ranges of these are both quite large? What does that overall trend look like? So this would be all the dots, including the gray ones in the back. And overall, we see when there's more water, there's more sediment. This is probably not that surprising. What having these range of extreme events, including Hurricane Irene in blue, which was closely followed by Tropical Storm Lee in darker blue, Hurricane Sandy in red, we wanted to see how did those extreme events compare to our overall range of variability. And we're seeing they're pretty much within the range. The highest discharges, are certainly measured during these extreme events. But those aren't even necessarily the highest concentrations of sediment in the water there. And the other thing we notice, we aren't seeing a major shift, and we've done more analysis on this. We don't see a major shift in this overall pattern before and after the hurricanes. That's something we wanted to know is, did we hit a new state of watershed? Did the functioning of the watershed change afterwards? We didn't see that here. Having done a compilation of a lot of these studies around Hurricane Irene, well, I can tell you that further north up in New England, they did see a state change and they did see a baseline concentration shift. But further south in the mid-Atlantic, that didn't occur. And there are some reasons about that. So we're going to call the mid-Atlantic area and our system here fairly resilient in the face of these hurricanes. But this is all fine and good and interesting. We want to know something about what makes up the material that's in all of these dots here, right? Is it that landscape surface erosion from those tilled agricultural fields making its way to the stream channel and out? Or is it that material coming from those really deep stream banks, right? There's potentially a lot of material there. They're very long because they run the whole span of the stream, and they're approximately one female scientist high. So this is a lot of surface area here. And this is really where we hit it with the chemistry. And so we use similar mixing models, in some cases similar to what I presented, with telling us if it was soil water, groundwater, or rainfall coming out of the stream. But we're doing similar things on the particles themselves. So there's all kinds of chemicals, including different ions and different isotopes or different varieties of the same element that have different masses because of different numbers of neutrons that hang out in the atmosphere. They come down in rain. And with that rain, they interact with whatever is on the surface, be it exposed soil, tree canopy, etc. And so what I particularly like to do, and what Lucy has started learning since she's come to work here, is use a suite of fallout radionuclides. These are radioactive isotopes, some of which, beryllium-7 and lead-210, are in the atmosphere naturally. So these are not produced by weapons testing. These are not produced by nuclear power. Cesium-137 is produced by those anthropogenic methods, and that's not the focus of this study. What we do is we use these beryllium and lead isotopes. We measure how much comes down in rain, how much is on the surface, and how much ends up being in the stream. And it's sort of like that color mixing project, right? If we take yellow and we take blue and we mix them, we get some sort of green. So we can think of the rainfall concentration or the rainfall chemical composition being yellow. And we can think of that composition that hasn't recently been touched by rainfall, maybe it's on the stream bottom, maybe it's really deep down in the soil profile, as being some form of blue. And what's coming out of our watershed is some mixture of these two. And we use chemistry and a little bit of mathematical modeling to tease that apart. So this means we're starting to understand variations in those rainfall concentrations to begin with. We measure lots of rain and measure its isotopic activities. And what we found just by looking at one of these tracers, beryllium-7, alone, is that there's a massive seasonal difference in its concentration and its flux deposition. The flux data isn't shown here, we're just focusing on concentration. And one of the major correlates that had previously been published was season. But looking further, we found that there really was a correlation not just among season, but also to type of storm. So if we looked at a convective thunderstorm, that chemical composition of the rain was really darn different than a hurricane. And it was really darn different than a frontal system that would be hitting that mid-Atlantic in the summer. And so by looking at this, and then also in conjunction, looking at these same chemical tracers on stream sediment, we're starting to build models that we can look apart and say what was recently in contact with rain, what wasn't recently in contact with rain. And did that contact come by rain hitting the surface water, directly, or did rain hit some sort of landscape field that would erode into the stream channel? And so I'll walk you through some of these mixing plots very, very quickly to give you the bottom line. So here we're looking at two different isotope tracers, one in the top panel, beryllium-7, and one in the bottom panel, which is excess lead-210. Both of them on the y-axis, we're measuring that isotope activity in the channel export. So this is on the suspended sediment, on that how green is my particle. Across the x-axis, we're looking at the amount in the rainfall that's hitting the channel directly. So this would be rain coming right onto the river and hitting all of the banks and deposits and everything that's in there. Both of them have one-to-one lines. So if our storm input versus output plots above the one-to-one line, we have some sort of net export, meaning we're either taking stuff that was stored in the channel previously, or we have a subsidy from our watershed landscape. And so we have erosion coming into the stream and making its way out. Conversely, below the line means we're exporting even less than what we're getting into the channel to begin with. And what you'll notice here is doing the same or a pared down version of the storm type analysis, the hurricanes are way above the line. So hurricanes, we're getting eroded material from our landscape, potentially making it in. However, thunderstorms, summer thunderstorms are below the line. We're not. Summer thunderstorms are really kind of pulsing material that's already in the channel through. And other frontal systems are close to the one-to-one line and a bit different, usually related to season and freezing conditions. A whole other suite of tracers related to the organic matter composition, carbon, nitrogen, and metal oxides is what one of the projects Lucy's been working on since she's been here. We're doing a similar sort of mixing model. So statistically, this is done with the principal components analysis, and there's all sorts of data from over 150 samples that go into this. But the punchline, so the baked cake that we'll take out of the oven here, is to look at a triangle of different groupings of sources. So we might want to look at watershed surface samples. So this would be surface erosion from the agricultural fields or the pasture surfaces. Or some of the small urban area within this watershed. The blue circle. The green circle would represent what's in the river channel. So this is what the stream bed is made up of and is storing. And all the way down here, this red circle is kind of subsurface material. So if you were looking deeper in a soil profile. So we have really deep road cuts in this region where you see a full soil profile just by driving to work down the road. We also have a lot of upturned trees, and so in a tree throw, it brings up that material that's not right at the soil surface and makes it kind of prospect for erosion. So what Lucy did was look at over 50 different storm samples and try to tease apart where is this particulate organic matter coming from using a bunch of chemical data and this mixing analysis. And what we found was really very interesting things. Under high flows, so like at the peak of those hurricanes, we're getting that mix of yellow and blue that we were expecting, where we're getting some material that was probably on the stream bed, and we're getting some material that's that surface erosion. But under lower flow conditions, so in more frequent rainfall events that might happen every month, every other week, or at the very tail end of the storm, you see these L's pulling way out of the mixing triangle. And there's a lot of detail on this that I know I'm glazing over here, but the paper's in revision, so hopefully you'll be able to read it soon, or we can talk about it. These lower flow samples are pulling out of those other sources. And what these do resemble chemically from a bunch of other data that we have is material that comes together in the stream channel itself through biogeochemical and ecological processing. So it's a lot of get together of mineral oxides coming in from groundwater and organic matter coming in from higher in the soil profile. And really quickly, we see these same dynamics happening throughout the hurricane over the course of the storm. So if we look at two storm hydrographs for Irene and Sandy, over the course of these discharge over time, we took really big samples at a couple of different time locations in the storm. And when we're in that big flood period, the pink samples are falling within our mixing triangles. The black, towards the tail end of Hurricane Irene, is pulling out and looking like that in-channel source. And we see that starting to happen with the second pink pulling out on the downward flood recession during Hurricane Sandy. So later in storms and under these more frequent conditions, it looks like it's really processes in the channel that are determining what is exported from the material. So this watershed really has kind of a tale of two conditions, even though it's the same system. Under high flows, everything connects up much more readily. So we're not limited by material from the landscape having water to bring it into the channel. And so it's more of contribution to the stream and landscape connection. There's an aspect of hydrology called hydrologic connectivity that's a very big research topic right now, and that's certainly what's happening here under high flows and during flooded conditions, and we see this too a little bit with frozen ground. However, the chemical tracers for sediment source confirm this, but under lower flows, what happens is we look like we have a really tight cycling between the rain that hits the stream, having the opportunity to mobilize material stored within the stream, and export what is biogeochemically and ecologically produced in the stream. So in short, chemical tracers allow for a much deeper investigation for the source and flow path connection of what's coming out of our watersheds. The same system can react differently to different weather conditions. And so it's, to a hydrologist, really important to understand what's going on with the water. Are we hitting a wet system? Are we hitting a dry system? Are we hitting a frozen system? Are we hitting an unfrozen system? And so at high flows, watershed landscape sources matter. Under lower flows, the story is what's going on in the stream, and it's the ecology and the biogeochemistry linked to those processes that really provides a bit of the driver. And so with that, we're going to talk about areas that definitely have frozen soils sometimes. Now I'm going to hand it over to Lucy to talk about our work in West Swan.
Yeah, so this system in West Swan, I'm very excited to show you this time series video that we have of the snowmelt dynamics that happened this past spring. It's the snowmelt and that sort of hydrology aspect is really incredible. I've never worked in a system like it. It's kind of overpowering. So I'm going to show some preliminary results of this study we're doing up in St. Louis County that's looking specifically at the effects of forest harvesting on water quantity and water quality in a bigger stream, river almost. And so just first a little bit of context and history. I think it's really interesting. In the lake states here in the Midwest, we have kind of a unique relationship between forest management and forest timber harvesting and hydrology, sort of different from what you would expect in the western U.S. or in the southeastern U.S., where they also have timber harvesting activities happening, but they also have a lot more topography, and so that affects the hydrology in different ways than it does here. Here we have really extensive surface water coverage, and combined with minimal topographic relief, it's really flat, and so that really affects how the water moves and the spatial extent of where the water can come from to contribute to a specific area of a stream, and so it makes us sort of unique in that way. And then when you couple that with timber harvesting, it makes for interesting relationships. We're fortunate that we have Marcel Experimental Forest here up just outside of Grand Rapids, run by the Forest Service. And they've done a lot of work over the past decades looking at the intersection of forest management and timber harvesting and hydrology, water quality and quantity, both in upland systems and peatland systems. So it's really applicable to our region here. A lot of that work was done in the 70s and 80s on the effects of harvesting and hydrology by Sandy Very, who used to work there at Marcel. And so we have this nice long-term data set to call on, combined with recent and ongoing work to answer these questions about, or to start to answer these questions about hydrology and forest management. And a lot of the work that has been done at Marcel has really been taken to heart by forest managers throughout Minnesota and sort of throughout the region in terms of driving how we view BMPs and the implementation of BMPs for water quality and discharge and things. So a lot of the work that was done at Marcel earlier has fed into what we know and what we understand, how we approach water diversion and erosion control metrics, stream and wetland crossings, how we approach those things during harvest, and then the establishment of riparian management zones and filter strips. So a lot of that work at Marcel earlier has fed into how we approach this and how we understand these implementations of these things. And when they are implemented, they're generally effective at keeping the stuff out of the stream that we don't want there. Okay, but one thing that that Diana and I got sort of interested in exploring a little bit further is this idea of this sort of 2/3 rule. That's what we call it, I guess. That's been broadly adopted, I guess, in terms of like the mentality of the approach to timber harvesting, certainly in Minnesota and maybe regionally as well. That came out of some work in the 1980s, the mid-80s that Sandy Berry did at Marcel in these small watersheds. And basically what he said was that when you've got a watershed where 2/3 of the area of the watershed approximately is either open land, it's been completely cut, or it's a grading, it's young forest. When you've got that proportion of a forested watershed in that condition, then that's going to substantially change the hydrology of your system such that the channel forming flows, the really important hydrologic events, are going to double or triple in size, right? And so that's like a nice clear guideline for timber, the extent of timber harvesting, right? And so it's attractive to adopt that sort of rule, that guideline. And it goes along with some other things that he mentioned in his paper, basically stressing that the management area, the size of the management, is sort of the main control on the land use impacts of water quality and quantity in a system, right? But the thing I think that is also important to emphasize is that he also mentions that the forest effects on water are not uniform. There's not a single right answer. So applying a single guideline to different watersheds or different forests across multiple scales might not be appropriate. And so that's kind of what motivated this work that we were doing in West Swan, where we're looking at, so it's in St. Louis County, just not too far from Marcel, so a similar system. But we're looking at a larger river system, this is the West Swan River here, and a huge watershed, much, much bigger than what they were working in at Marcel. At Marcel, it's maybe like 50 or 100 acres size watersheds, so headwater. These watersheds are much, much bigger, and the whole system is much, much bigger. So you can see Hibbing here, and our site is maybe like 15 or 20 miles south of Hibbing. And we've got at this site an upstream, we've got an upstream site and a downstream site, and then the timber harvest occurs right in the middle of those. And so I guess I wanted to keep this large scale map just to emphasize the relative size of the watershed and the harvest area. And you can barely see the harvest area in here. It's this little black blob right in between our two sites. You can see it better here. It's 100 acres. So it's a good size harvest at an upland site during the growing season, but still in this context, in the setting of this huge watershed. And this is sort of representative of what you might see across northern Minnesota in terms of, you know, the setting for a timber harvest. This is what we wanted to look at in terms of, you know, what might we see potentially in terms of the effects of this size of a harvest right next to the stream in terms of the water amount and the water quality coming out after the harvest. Okay? And we're using our upstream site here as a control because we're not expecting this area here to be affected by the timber harvest because water moves downstream. So like I said, these watersheds are huge, 78 square miles and 80 square miles for the upstream site and the downstream site respectively. And our harvest area, relatively speaking, was much, much smaller than this 2/3 guideline, of the watershed. So 100 acres happened during the growing season, in an upland aspen stand, pretty close to the river. You see though, there's this space here. They left a nice like riparian buffer there. They didn't go right up to the stream edge. And this harvest was completed just a few months ago in early September. And it was pretty extensive. They came in and they took everything. And so now we're going to spend the next year measuring the post-harvest data and kind of see what, if anything, the effect of this harvest is. So like I said, we established 2 sites, an upstream site, immediately upstream of the harvest area, and a downstream site immediately downstream of the harvest area, both on the West Swan River. And we wanted to think about what a growing season upland harvest might do to the water quantity and quality at our downstream site. And we're thinking of potential changes in the magnitude and the timing of things like discharge, dissolved organic carbon concentrations, because DOC is really important in the waters here. These watersheds typically have a lot of wetland coverage, and they just sort of like hemorrhage dissolved carbon. It's kind of hard to see. I don't know if you can tell, but you know, this water is brown. It's very brown, and it's brown year round. It's like tea because there's so much dissolved carbon moving through it. Also looking at total suspended sediment, TSS, like Diana was talking about earlier, and a related metric, turbidity, so water clarity. And so we started collecting pre-harvest data in the fall of 2016 at both sites, and we're going to continue for the next year now that the harvest has happened. We have thrown a slew of monitoring equipment and water collection equipment at this project. At both sites, we have deployed turbidity probes in the stream, sensors that measure water depth, temperature, and conductivity to kind of give us an idea of the ion concentration in the water. We've got a sensor that measures continuously the discharge at the location in the stream where it's put in. And then we have these automated water samplers that are pretty big. We keep them in, well, you can see here this 55-gallon drum. That water sampler is inside that drum, and it can hold 24 1-liter bottles. And we can sort of like program the water sampler to collect water samples at different intervals, or, you know, pacing with the different flows and things like that to get at different flow conditions during a storm or seasonally or whatever. So we've got all of this stuff for the most part at each site. One thing I really want to emphasize, and you'll see this in the video that's coming up, is that the discharge variability here is really huge. And so this is not the West Swan. This is just a tributary that goes into it. And when Diana and I were scouting locations to do this study, we were walking around and, you know, came across this tributary. And it was remarkable that there's very little water here. This is early growing season. You can tell by the leaf out here that there's very little water in the stream right now. But just a few weeks earlier, when the snow melt was really going, the water was, you know, would have been above Diana's head, because we know this because of these sediment deposits that were on the top of this log. So there's so much water moving through, carrying so much stuff that, you know, when the water receded, all that sediment was deposited on that log. So it's just a crazy system. I've never worked in a place like this before. Sometimes maddening. So I'll show you a time series of the spring snowmelt from earlier this year. And it's about a 3-week interval here, and you'll see just how crazy it gets. And it's, if you watch the left-hand side of the video kind of closely, you'll see what this snowmelt flow did to our big 55-gallon drum automated water sampler. This is the maddening part. Okay, so how do I now?
Spacebar.
Yes. There we go. Okay, so still icy. This strap here is ratchet strap that's holding down our barrel here. We have a game camera that is focused on a staff gauge, so we can also measure water level using that, sort of an analog way, which is nice. So you see the snow melts, and then a little bit more falls, and then eventually the snow melt really begins in earnest, and you see the water level start to come up. And then it really starts to come up. It really starts to come
Time
up.
period from when it started rising to, let's say, now, are we looking at?
A few days. Well, so the whole thing is about 3 weeks. So I would say like a week. Yeah, so you see that we had a barrel, and this is the battery, the car battery that that powers that automated sampler, and you can see the battery case is covered with sediment, right? So thankfully, our water sampler was plugged into the battery, and so there were cables and stuff that tethered it, so it didn't, I mean, otherwise we would have seen it floating downstream. But I mean, that water sampler in that barrel was full of 24 liters of water, like it's heavy. But I mean, this stream is just like whatever, you know, it's a really dynamic system. And so sometimes it wreaks havoc with our instruments. Okay, so now to get back to this, I just click out here, is that right? Yes, all right. Okay, so I'm gonna show you some of the data that we've been able to collect over the last year, and show you some of the variation in flows. So you're familiar with this. This is precipitation from October of 2016 to October of this year. So basically a full year of data. And these are daily total precipitation. And so we have, you know, some storm events that give us quite a bit of rain in a very short period of time. A lot of variability in precipitation. And then this red bar here is showing that 3-week interval that was on the video. And so you can see that there wasn't a whole lot of snow or rain coming in, but this interval coincided with the transition from below 0 temperature. So here I'm showing air temperature in Celsius, and this line across the middle is 0. So below freezing temperatures, average annual temperature, to above freezing, and this is where the snowmelt happens. And when we get that snowmelt, you see this is stream depth in meters. And you see that the biggest stream depths, and this is also similar, like the biggest discharges, happen during this snowmelt event. And so, you know, at its peak, the peak depth was close to 18 feet, 6 meters, right? And as we get further and further into growing season, the stream depths and the discharge kind of tail off, and then as the vegetation kicks in, we really see an effect of it. So even when we have big rainfall inputs, big intense storms, the stream level doesn't really come up nearly as high as what it does in the snowmelt period, and the discharge shows similar patterns. So really a stark contrast between dormant season and growing season in terms of the hydrology. And so it'll be interesting to see, now that 100 acres of trees are gone, you know, whether we see the inverse effect of that lack of vegetation, I guess. So here, this is the last axis I'll add here. Over here, I'm showing DOC concentrations, dissolved organic carbon, in milligrams per liter. And you see a lot of data points stacked up on top of each other. These were samples that we took with our automated water sampler during individual storm events. And so if you kind of zoomed way in on the time scale, then these points would spread out and we would be able to see kind of temporal patterns during storms. But what I want to show this way is just that, first, there's a lot of variability during some storms. So a huge range of concentrations, you know, almost a doubling on some dates from 40 milligrams per liter up to 80. So a lot of variability just within an individual storm event. But also seasonally, there's this very obvious pattern where during the early growing season, dormant season, we have low organic carbon concentrations in the stream. And then as we get more and more into the growing season, that vegetation, the wetlands particularly, really kick on and just start leaking organic carbon into the stream. And so this is at the downstream site. The upstream site in yellow here, it just almost exactly overlaps. So DOC-wise, these two locations are very similar in the pre-harvest period. It'll be interesting to see what it's like in the post-harvest period. And then similarly with total suspended sediment concentrations, we don't see at the downstream site here in green, we don't see really a seasonal pattern at all. And we see not as much variability for the most part during individual events, with the exception of like a couple really big spikes here and there from 1 hour or another. But generally lower, you know, lowish concentrations at the downstream site, whereas at the upstream site we see a lot more variability within individual storms, and just overall higher concentrations of sediment. And I'm not exactly clear on why there would be such a difference right now, except that maybe, somewhere between the upstream site and the downstream site, maybe the water flow slows down a whole bunch and things just sort of settle out. But I'm very excited to see what happens in the post-harvest period with these downstream numbers, to see if they either start to approach what we see in the upstream or surpass them. And that's related to some of the other stuff that we're going to be doing next. So first, we'll continue measuring discharge and DOC and TSS and turbidity at both sites through the summer of 2018 to really capture the post-harvest period, or the early post-harvest period. And then we're going to start doing, similar to what Diana described for the work in the eastern U.S., we're going to start doing these chemical fingerprints of both the suspended sediments in the stream and also potential sources throughout the not the entire watershed, but the near stream area. So we'll be collecting soil material from, for example, stream banks, or in the channel, the stream bed, or like sand or sediment bars that occur. Certainly soils in the harvest area, and then materials from some of those smaller tributaries that clearly transport sediment, as we saw with that picture with Diana. So we're going to be collecting those materials and doing that battery of chemical and isotopic analyses to try to match up the chemical fingerprint of those potential source materials with the chemical fingerprint of the sediments in the stream, and see if we can describe the sources and how they might vary under different hydrologic conditions. And something that I'm personally interested in, I know that Diane is interested in, this sort of event scale, storm event and snowmelt event scale dynamic. We're going to be delving more deeply into the hourly and sub-hourly samples of water and sediment that we've collected, and really trying to understand how the biogeochemistry of the stream water and sediments varies with the discharge conditions during individual storm events and snowmelt. So that's all I've got. Think
we have a lot of people to
we have
thank,
a lot of
right?
people to
Like
thank.
It takes a village.
Yeah.
So the White Clay Creek work was funded mostly by NSF, and continuation of it is still funded to us from the Delaware Watershed Research Council and William Penn Foundation, as well as our collaborators that have all contributed to this work at the Stroudwater Research Center, University of Delaware, and University of Exeter in the UK. The West Swan work is basically all hands on deck at some times within the Watershed Hydrology Lab here.
Everybody
in our
has to make a trip to the field.
Yeah, like more often.
If you ever want to go up north, just let me know. I'm happy to have people come help.
So Ethan, Mark, Zach, Tate over the summer, and Hannah. We're working in conjunction with Quentin Legler and Cheryl Adams at UPM Blandon, because the whole study was set up to monitor a kind of business as usual harvest that UPM Blandon just kind of conducted per their normal procedures. And that's been funded by the Minnesota LCCMR and the National Council for Air and Stream Improvement. So, questions?
Now this watershed, between the upstream and the downstream, the only change has been this land. It's isolated enough that the only change has been the harvest.
Yeah, so
roads that are there, or there's other things.
No, there haven't been, new roads or anything like that. And so it's kind of interesting that we wanted to look at an upland harvest, right? And so there's this little pocket very near the stream there of upland. And then as you get further away from the stream, it becomes wetland. And so that hasn't changed. So, yeah.
So that's my question. I'm wondering to what extent the hydrology of the stream is a function of that sea of woody brushland that it sits in. Our work in brushlands has suggested that they're super flashy and they can change by like 1 to 2 feet on a day-to-day basis. And I'm just curious about the role that brushlands might actually play in the stream, and whether you are going to sample those as a potential source as well.
Source, yeah. So I wish, well, Lucy too. We wish we had more money to sample more sources in this watershed.
This is
always
exactly
the case, right? Like money and data, we need more.
And this is exactly what you proposed as sort of an extension of this project
Yeah.
for the last round of LCCMR funding, was to look at exactly that, extend it more into the wetlands and kind of try to capture
Yeah,
that.
there was a brushland, like a lowland brush,
Woody wetland.
lowland woody wetland that's up for harvesting soon, that's networked in along one of those tributaries, and that was not recommended for funding to move forward. But I think a big part of the woody wetlands isn't just the amount of water, but that's clearly linked into the dissolved organic carbon. Because I mean, this is brown water, right? And just to look at, quantify the sheer flux of carbon coming out of these watersheds, because you're looking at, we don't have all the discharge rating curves constructed yet, but when the water is 6 meters deep, it's still running. What's low for this stream is brown, and there's 20 plus milligrams of carbon per liter of water.
That's the lowest measurement. That's on the low side.
Per liter. So crazy. Multiply concentration by the amount of liters of water that come through there per year.
We can do some field work exchange. You guys can come tromp around in the woody wetlands up there with us, and we'll help you with the stream.
Gladly. I would love it.
Survey stream channels.
I would love it. Yep.
We have plenty of waders.
Yep, we do. We do. And all the holes are patched in them, so.
As of now, yes.
Right now.
So what does the literature tell us about like the upstream, downstream, about when you might see some normalization of the flows and turbidity as sediments after a harvest? Is that a period?
So honestly, in rivers this size, that literature is kind of non-existent.
Really?
So a lot of the literature that was developed specific to forest harvest is in much smaller watersheds. And so they're dealing with shorter flow paths, right? Shorter time for water coming from the uplands to make it out of the stream. The literature currently says in Minnesota, it's like a 10 to 15 year sort of recovery time interval on the discharge. The sediment could be different. It could be more or less depending on the region of the country you're in. But those are all done on those really small systems. And so when you're in bigger rivers, there's a longer variety of flow paths that water could take to get to where they're coming out of our downstream site in West Swan.
And the sources might be different. It might be more in-stream stuff. And so it's just the sheer increase in water, not necessarily more stuff being transported in water, but just more discharge
itself.
We're, working stuff up that's already in the stream. We'll see.
It's really kind of, it seems like bigger rivers and much smaller streams are studied. Yeah. And we were really trying to go in and not dictate a harvest, but work with Blandon and say, where are you harvesting, that we can look in either 2 adjacent watersheds or above and below. Yeah. And so to work with that harvesting pattern to get this business as usual harvest scenario, this was kind of our best option to get an upstream and a downstream and a sizable upland harvest. Yeah.
Is there a management component, a land management component to the Pennsylvania work? I'm not seeing that.
So there's not as much of a change in management component. There is a big question about what influence the agricultural land versus the reforested land has on what's coming out of the stream. So when does the agricultural land contribute eroded material versus the forested land? But there isn't a change in state of the management over the course of the time. So
I was just looking for management implications for the work there. For example, in the Minnesota River Basin, they do a lot of this type of work that has a very management orientation as to what, after they've characterized it, what do we do about it?
Right.
Quantitatively.
So what we're seeing in the Pennsylvania work, and I'm affiliated with other work and sort of more watersheds in that region that would then drain to the Chesapeake Bay. And so there's big questions about best management practices in general and how long might it take their effect to be seen downstream, right? And a lot of the best management practices are things like stream side buffers, riparian buffer vegetation, which we also work with some here in Minnesota. And so there, the implications are really different in terms of if the big story is processing in the channel itself, we need to think about best management practices surrounding that versus putting riparian buffers in place that serve to disconnect the watershed sources from the channel. But if the sediment source is in the channel to begin with, it's sort of like, you know, putting a bandage on your arm to fix your broken leg, is the way I describe it. And so really thinking about the conditions in which it's within the channel itself versus a landscape field contribution. And so there's some of that that resonates with the work that's being done in the Minnesota River Basin, but what's particularly where it's amped up in the Minnesota scenario is with all the modification of water flow paths that's going on in the agricultural lands. And so there's a lot of research involving what that does to the hydrology alone. So it's not just a matter of the sediment source, it's a matter of increasing the flashiness of the stream, increasing those spring discharges higher, and what that can do for channel erosion in the bluffs and bed and things like that.
And the patterns of ET with different land uses. Your rate, quantitatively, how does your ET affect that under different scenarios, cropland versus?
Oh, sure. And any of the work that I'm familiar with and been a small part of with other students is thinking about when you're looking at a lot of stochastic modeling for how long it takes different water flow paths to make it through the basin, accounting for ET, not just crop versus forest land, but kind of the phenology of ET, Rebecca. How does ET change throughout the course of the year when you have corn versus fallow versus if there's a cover crop implemented, what that does? Yeah, Ray.
I was curious about the data you did show, because it kind of is an area that I work in. You said something about 16 RNA genes and sequencing and some
so
work, and you didn't mention it.
Yeah. So we have like millions of dollars of data. I couldn't fit it into a 20-minute talk and then give you all a chemistry test at the end. So I have a collaborator who's still at the Stroud Water Research Center that some of my big hurricane samples and all the intensive samples I took for the 2.5 years we were actively sampling there, he took subsets of those and analyzed them for DNA signatures for the microbes that are attached onto the suspended matter. And so we're working through that data now as well. So it had the DGG, okay, I'm not a microbiologist, but it had different levels of DNA sequencing, including high throughput. And so just an astounding amount of data that he and a student there are working through. And what they're finding, we've talked about this past spring, is really, really consistent with Lucy's findings on the POM and mine with the fallout radionuclides, is that over the course of a storm, so he needed such little material that we could do like hourly through a hurricane, we're seeing this progression of sources throughout the course of a single storm, where a big component of them on the tail end, I believe, and when the flows are lower, look like in-stream bacteria. And so able to tease apart what are the kind of watershed source area attached bacteria DNA signatures from what's in the water column itself. And so we're kind of hitting it with a chemical and a microbiological tool, and they're all consistent with when is it the upland, and by God, there's something going on in the stream.
And with that, just to stay on time, let us